Welcome to the Ask the Expert. Today we are speaking with the team at Levicure, coming to us from Tel Aviv. They're talking to us about some work they've done, which, is the title of the talk is Type 1 Diabetes Remission and Beta Cell Regeneration, induced by oral administration of triple drug combination of the DDP4 inhibitor, PPI and GABA in NOD mice and human patients. So we have Dr. Shmuel Levit. He's the founder and CMO. He's an MD PhD with over 35 years of clinical practice. He's the head of endocrinology at Diabetes and Metabolism Institute at Suda Medical Centers in Tel Aviv. We have Daniel Koshalev. Daniel. He's the co-founder, CEO, and strategic management director. He's the former CEO of Health Ingredients Company. And we also have Mike Teeler. He's a strategic advisor who has a bachelor's in pharmacy and pharma evaluation, and he has a long career in pharmaceuticals. And finally, Lucy Koshalev. She's the co-founder and general counsel, and she's going to introduce the concept. I did want to bring up the fact that Dr. Levit heads up the Endocrinology and Diabetology Institute at Suda Medical Centers, and he's also a leading specialist and endocrinologist and expert in the treatment of diabetes in this area. So welcome the team from Levicure, and I'd love to hear more about what's going on with your approach.
Thank you so much, Monica, and it's a pleasure to be here. So hello, everyone. Thank you for your time today. My name is Lucy. I'm part of the Levicure team. We're a small startup in Israel that was founded by Professor Shmuel Levit. So Professor is first and foremost a clinician with 35 years of experience treating people with endocrinological disorders. Within his own private medical practice, he was able to establish that a combination of two well-known FDA-approved drugs and a supplement GABA can lead his type 1 diabetic patients into remission or significantly reduce their insulin demands. So Daniel and I helped Professor Levit set up a company to protect and push his discovery forward. And Mike has joined Levicure shortly and brought on board his incredible pharma expertise. Thank you. Next slide, please. So you must be wondering, what is the combination of these drugs? It is DPP4 inhibitors such as sitagliptin, a proton pump inhibitor, in our case it's omeprazole, and a neurotransmitter GABA that is largely sold as a supplement without prescription over the counter. So the combination of these components is not random, and in 2017 GABA was actually found to induce the conversion of alpha into beta-like cells. The double combinations have extensively been researched for years, and for instance, that same year, 2017, the combination of GABA and sitagliptin was shown to promote beta cell regeneration in mouse models, and later on in transplanted human beta cells and streptosin-induced immunodeficient mice as well. So in 2020, it was also discovered that GABA and GLP-1 receptor agonists work together to inhibit the expression of TXNIP in pancreatic beta cells, which promotes beta cell proliferation and reduces apoptosis and damage. So in separate studies for decades, PPI was also extensively researched, specifically by Alex Rabinovich and his team, most notably with a combination of DPP4 inhibitor in 2009. On its own, proton pump inhibitors were repeatedly considered for type 1 diabetes treatment, and in 2011 reported to improve glycemic control by increasing gastrin levels and blood in the pancreas. Unfortunately, however, all of these results were made in animal models and cell cultures, and these findings did not successfully translate into humans. And here is where Professor Levit managed to combine these separate three components of extensively researched drugs. And while most discoveries begin with mice studies that translate into human data, in our case, Professor Levit was able to see the effectiveness of the triple combination on 19 of his adult human patients first within his own clinical practice. So the results are really impressive, with 6 patients out of 19 achieving complete remission and getting off insulin. And an overall 60% of the group managed to significantly lower the insulin demands. So in order to secure the patent for the triple therapy, we had to show a proof of concept animal model study. In our case, it was NOD mice from Jackson Labs comparing the triple combination with all the double therapies, including those previously researched, and the placebo. So the results corresponded really well with Professor Levit's findings on humans. And based on our observation analysis of the existing research, we were able to form a hypothesis on the mechanism of action, which I'm going to briefly cover in a minute. I have to say that no additional experiments have been performed yet to support our hypotheses, and we're currently collaborating with Uppsala University and Alex Urbanovich as well to publish our findings and continue the research. And we'll be more than happy to engage in other academic collaborations to further our research on our hypotheses. Thank you. And next slide, please. So on this slide, you can see our patent applications. We have applied for a number of patents that are covering our therapy in different ways, including the neurodegenerative disorders. As of today, we have successfully obtained a patent for the treatment of type 1D in the US and are in the final stages of getting a patent for triple therapy in the EU. Thank you. Next slide, please. So I'm now going to briefly cover our hypotheses on the mechanism of action. As a foundation of our hypotheses, we need to start by addressing that the endocrine cells of the pancreatic islets and the neurons of the central nervous system have many features in common. They express the more genes and cell surface receptors, glutamate receptors, and GABA receptors, and most importantly, are capable of releasing neurotransmitters such as GABA. So within the framework of our triple therapy, we are concentrating on glutamate, GABA, and GLP-1 receptors. We believe that there are two key dysfunctions that play an important role in the onset and development of type 1D. Both of them have already been clinically documented. So the first one is significantly elevated glutamate levels, and the second one is overexpression of serum DPP-4 enzyme. So elevated glutamate levels are causing beta cell dysfunction and damage, and later on apoptosis through overactivation of glutamate receptors, and also induction of oxidative stress in the cells, and in parallel greatly affect the proliferation and behavior of the immune cells. So the DPP-4 overexpression type 1 diabetes development is problematic through its enzyme cleaving activities of the GLP-1, and also as an activation marker for the immune cells. Thank you. And next slide, please. So in this slide, we have tried to show some of the molecular interactions that play a part in type 1D as we see it. And as I already stated, we think the combination of GABA, sitagliptin, and omeprazole seem to safely and effectively target some of the key dysfunctions that can be potentially caused by excessive activation of serum DPP-4 and elevation of glutamate in immune endocrine cells that happen in development and autoimmunity of type 1 diabetes. So glutamate on its own is extremely important as it modulates the secretion of glucagon in the alpha cells and insulin in the beta cells. However, elevated glutamate levels can accelerate the beta cell dysfunction and lead to cell death through excessively activating glutamate receptors and severely affecting the neuroimmune function of the T cells, as is already evident in a variety of autoimmune neurodegenerative conditions, such as multiple sclerosis, for example. So GABA counterbalances glutamate, has a key role within the islets in the regulation of islet cell function and secretion of insulin. And beta cells are some of the few cells, including neurons and interestingly enough, B lymphocytes, that are able to convert glutamate to GABA with the help of GAD enzyme. As you are aware, GAD antibodies are present in type 1 diabetes and in various other neurological disorders and interfere with GABA transmission, blocking GABA production in the endocrine cells and neurons and possibly contributing to glutamate-induced excitotoxicity. So as GABA is produced and secreted by the beta cells, it is suppressing glucagon secretion in the alpha cells. And through positive feedback loop, it's also absorbed through GABA-A and GABA-B receptors in the beta cells and has been shown to promote beta cell regeneration and proliferation. It is important to note once again that GABA induces the alpha to beta-like cell conversion, which is extremely important for increasing the beta cell mass during the treatment and restoring the pancreatic function. Also, under the condition of oxidative stress, for example, caused by glutamate elevation, GABA was found to need the GLP-1 receptor to inhibit the TXNIP, which is effectively a glutamate-dependent precursor for glutamate excitotoxicity. So the more TXNIP, the more the oxidation stress inside of the cell. This means that we need the GLP-1 receptor for GABA to effectively mediate the oxidation in the beta cell and prevent damage and apoptosis. So the GLP-1 is not surprisingly an agonist for GLP-1 receptor, and it is cleaved by the DPP-4 enzyme. If there is an elevation of DPP-4 and overexpression of it, it will substantially reduce the availability of the GLP-1 that is needed for insulin production and management of glutamate-induced oxidation stress. So the DPP-4 inhibitors inhibit approximately 90% of the DPP-4 activity, leading to at least a twofold to sometimes threefold elevation of the GLP-1, and therefore prolonging the stimulation of GLP-1 receptors, activating the key transcription factors for better cell growth and survival. So in addition to this, sitagliptin also has anti-inflammatory properties that may be key in addressing type 1D autoimmunity. For example, inhibition of DPP-4 leads to decreased T cell activation, proliferation, and migration, as well as increased GLP-1 mediated uptake of GABA by immune and endocrine cells alike. So it was recently found that sitagliptin treatment potently inhibited membrane DPP-4 activity, but it did not affect either acidic and profile or cell viability in activated T cells. And this is extremely important because it means providing a narrowly targeted immunomodulatory effect, unlike the general immunosuppressants. So lastly, omeprazole increases endogenous gastrin levels, which stimulates better cell regeneration and improves glucose tolerance. So the gastrin levels are possibly elevated through activation of gastrin-releasing peptide, which is also one of the GLP-1 secretion pathways from the L cells of the intestine. And it has been previously reported that the combination of gastrin with GLP increases the beta cell mass and restores normal glycemia in diabetic mice. Thank you, and next slide. Furthermore, omeprazole could simultaneously increase the insulin secretion by inhibiting the V80-PACE proton pump in the beta cell insulin granules. So V80-PACE is a major protein expressed in specialized compartments of specific cell types. And in pancreatic cells, V80-PACE dependent acidification is important for insulin secretion. And it was recently found that V80-PACE blockade improves insulin secretion in diabetic rats. So this is a brief overview of the mechanism of action we're relying on, and we'll be happy to address any questions at the end of this presentation. And for now, that you are familiar with our mechanism of action hypotheses, after this brief overview, I'd like to pass over to Professor Levit to cover the human data we have obtained. Thank you very much.
That's great, Lucy. Thanks so much. And I see that you guys presented this at ATTD as well in February. So that's 2020. So that's very interesting that it's been out there. And yeah, I'd love to hear from Dr. Levit.
Yeah, hello everybody. Thank you, Lucy. So this is the clinical trial, pilot trial, which already was reported in ATTD in February 2020 and dedicated to the effect of triple therapy, namely DPP-4, PPI, and GABA in type 1 diabetes patients. Please, next slide. Okay. So we started on October 2018 all of the files of triple therapy patients in our clinic. There was 56 files of type 1 diabetes patients who were treated with triple therapy. Of them, only 19 patients fulfilled the protocol criteria. So we picked them up and we proceed with the statistics, and we compare the relevant metrics of parameters as before and after triple therapy. Next slide, please. In order to confirm that we are dealing with the clear-cut type 1 diabetes, and given our knowledge is that there is no pathognomonic sign of type 1 diabetes, this is the complex of signs. So we developed the type 1 probability assessment, which contains these 10 points listed here in this table. And we agreed that the patient who has 5 more points diverse the diagnosis of type 1 diabetes, and this diagnosis is justified. So next slide, please. So it is quite interesting to remark that the average score of our 19 patients was 7.4, but the score of most successful patients who actually stopped their insulin injection further, we are talking about 6 patients. The average score was even higher, 8.0. Also, the statistical difference, there was no statistical differences because of the small group of the patients. Next slide, please. So we are talking about 13 men and 6 women. The age of the first visit in our clinic was 48.2 years. Age of diabetes manifestation was 40.7 years, and diabetes duration on the start of triple therapy was 90.9 months. So there were patients who with the recent diabetes, but also there were patients with advanced diabetes as well. So observation period in our triple therapy was just a little bit more than 1 year on average, namely 56 weeks. Next slide, please. As for dynamics of daily insulin dose or daily insulin demands, we saw and noticed the highly significant reduction of the insulin demands in all our 19 patients. And the reduction was almost 60%, as you see on this graph. Next slide, please. The same is true regarding the A1C, which was reduced simultaneously with the dose reduction of insulin by 24%, and the difference was highly significant. Next slide, please. In order to show that we didn't starve our patients and didn't intervene with any diet possibilities and facilities, and we didn't use any other especially nutritional interventions, we demonstrate here that the BMI of our patients didn't change at all and remained stable throughout the study. Next slide, please. So all of our patients received the CGM investigation before and during the study. And regarding the time above the range, the hyperglycemia time in range, it was also reduced by 24%. The difference was significant. And this is the clear-cut sign of the successfulness of treatment. The next slide, please. I just want you to pay attention that we are talking about the range of 70 to 140 mg%, which is actually the range of the healthy person, and not from 70 to 180, which is agreed for diabetes patients. So it was a little bit narrowed and was compatible with a healthy person, as I say. Regarding the time in range, normal glycemia time in range, it was no difference, but it was the clear-cut propensity to elevation by 10%, but probably because of small groups of patients, we didn't reach the significance from the statistical point of view. And next slide, please. And there was a significant propensity to elevation of hypoglycemia time in range. By the way, it also wasn't different from statistical point of view, but it is worthwhile to say and emphasize that the hypoglycemia was the most desirable side effect of our therapy, actually providing us with the possibility to reduce the insulin doses and reduce the doses until the discontinuation insulin injection in some patients. Next slide, please. So this is the table which actually reflects the most successful patients number. These are 6 patients, as you see in the left column, and the lower row is the average. So we're talking about the age of patient, duration of diabetes, and time spent without insulin. You can see that we are talking about mostly the recently ill diabetes patients. Only one, actually patient number 18, only one patient had his diabetes for 11 months before he started the triple therapy. The others started the therapy no later than in 2 months from the beginning of the disease. And the duration of diabetes on average was 3.2 months. The time spent without insulin in all of these patients was almost 2 years on average. And the right column, you see that at the end of the 2020, we already had the 4 patients who remained without the insulin injection with the very good diabetes control. There were 2 patients who stopped the triple therapy from different reasons, and they unfortunately returned to insulin injections. Next slide, please. So in order to demonstrate the successfulness of our treatment, these are some CGM investigation of our patients, where again, in green, we see the normal range of blood sugar from 70 to 140. This is patient number 18 after almost 2 years of treatment and almost 2 years free of insulin. He is in a full remission, 95% in range, and it is compatible with the hemoglobin A1C of 5.3. Next slide. Another patient is in a remission for 32 months. Again, 99% in range. The estimated A1C is 5.4. And next slide, please. This is patient number 4 of our study. His remission lasts for 3 years, and he's 78% in range, and it is compatible with the estimated A1C of 6.0% without insulin for 3 years, as I previously said. To conclude, this pilot study demonstrates a high efficacy of triple therapy in type 1 diabetes patients. The insulin demands were reduced almost by 60% in parallel with highly significant reduction of A1C and without weight loss. 6 patients entered a long-term remission and became insulin-free. Those patients who had a tendency to higher type 1 diabetes probability score were most successful, and they also had the shorter diabetes duration. The treatment is safe and side effects seems to be mild and rare. And all in all, this study is a proof of our concept. Thank you very much.
That's great. Thank you very much. And then was Daniel going to say a few words, or would you like to, are we going to take some questions?
Yes, I just wanted to quickly go through the mice in Audi trial, which followed the Professor Levit human experiment, and we'll try to go quite quickly. We had a couple of arms, and the reason for we actually did this experiment was to confirm that it's the 3 components together that work synergistically, which provide this effect, not the 2 in placebo-controlled experiment. So we purchased an Audi mice from Jackson Labs. We took the female mice because they have the highest probability of obtaining T1D. At the start of the experiment, the age of the rodents was 17-18 weeks, so we started to treat them simultaneously all at once. Number of mice was 51 because we have different arms. We had a prevention group and we had a remission group. So this, we're talking about the experiment with the remission group, where all the mice was with the confirmed T1D status at the start of the experiment. We used 3 drugs as Professor Levit used in his human trials. We used GABA, we used sitagliptin, and we used omeprazole. All the mice were divided into 5 groups: the triple combination, all double combinations, and the placebo. The duration of the experiment was 10 weeks. We looked at the major parameters of the type 1D diagnosis: blood glucose levels, ketones in urine, body mass, and ketones in blood. We also looked at specific metrics like C-peptide, HbA1c, insulin, and glucagon, and we did fluorescent immunohistochemistry in the end of the experiment. To confirm the diagnosis of our mice with the type 1D, we set a strict criteria. So it should be 2 repeated measurements of the blood glucose higher than 13.9 millimole per liter. And for the diabetes remission, we used the similar criteria, but just the opposite. So it should be 2 repeated measurements of blood glucose below 13.9 millimole per liter, also accompanied by insulin discontinuation. Because we looked at our experiment at severely diabetic mice, and you will see it by the blood glucose levels, we were using continuous insulin, though exogenous insulin treatment for those mice at escalating scale depending on the severity of the disease. All the groups, as we said, all the mice were divided into 5 groups, and you can see here that they were divided homogeneously. And the treatment was dosed orally in the morning and in the evening in accordance with the amounts which you can see in this table. So our results very well corresponded to what Professor found in his human trial. The first thing is also the mass. So we didn't start our mice, and you can see that all the lines which you can see here, they're nearly the same, and there is no differences between all of the groups during the study in terms of the mice weight. The first major parameter which we're looking here is also median blood glucose levels, and you can see that the orange line, which represents the triple therapy, is different from all the double combinations and the placebo. It decreases during the study, and the significance becomes evident from the week 7. The significance of the difference between blood glucose levels is not only between the ABC and placebo, but between ABC and all the groups of our experiment. So the next thing which we wanted to show you is the division of the mice into different groups by the criteria. So you can see here mice in different T1D status of healthy mice. So red represents severely diabetic mice. So it is mice which by our scale were dosed with exogenous insulin injections every day, and we call them severe type 1D mice. Yellow represents mice with which we state as a mild diabetic mice who doesn't require everyday insulin injections, but their blood glucose levels are significantly elevated. And green represents healthy mice. So if we look at the placebo group, we can see that the red area increases during the experiment, reaching around 80–70% of all mice being not just with the status of T1D, but being on daily insulin injections, which could be also seen by the cumulative insulin intake, which increases daily and has a certain reaching 503 insulin unit by the end of the study. In ABC, it is the opposite picture. So we can see that the red group narrows down to only one mice in the end of the experiment, which obtains any additional insulin injections. And the green group, which represents healthy mice, increases to 55% of all mice in remission. And the amount of insulin injections flattens in the end. And the difference is nearly twofold between the ABC and the placebo. None of the double combinations were able to reproduce these results and provide any significant number of healthy mice during our study. To confirm that this blood glucose improvements was to improvements in the beta cells regeneration, so beta cell functions, we look at the C-peptide levels, and you can see again that the ABC, the triple combination here, is significantly different from the placebo, and it is the only group which shows the significant difference. And we can see the elevation from the beginning. So the C-peptide level increased tenfold during the study in the triple combination group. The same goes in the insulin and blood plasma. As you remember from previous graphs, the triple combination was the group which received the lowest amount of exogenous insulin injections. But in terms of the analysis of insulin and blood plasma, it showed the biggest results. It also increased tenfold from the beginning. It was the only group to show any significance with the placebo. Last but not least, it is insulin glucagon index, which is also significantly different between the placebo for the ABC group, showing that the insulin glucagon balance was restored for the proper function of the alpha and beta cells. Finally, we want to show you the fluorescent immunohistochemistry. You can see here that insulin is represented by the red, glucagon is represented by the green area, and blue is DAPI. The first mice is healthy mice. You can see a lot of insulin secretion and separate secretion of glucagon. The next mice is severely diabetic mice. You can see that the insulin is poorly secreted. And now we move to the group of the triple therapy. So the first one, it is the mice which converted from the high diabetes status to medium diabetes status, so not requiring any additional insulin injections. And we think of this group as a conversion intermediate group of alpha into beta cells. And you can see here the area which represents orange. It means that at the same time, the same area produces glucagon and insulin. And on the right, you can see the mice in the remission, which basically have the similar pictures as the healthy mice. In terms of additional statistics, we're going to do some additional evaluations of the monogistic chemistry. So for now, we have these visual representations as additional proof of our hypothesis. So to summarize, in terms of what we have seen in our DMS trial, it's again confirmation that our triple combination, which we have seen in human trial works. It's only the three together which can provide this unique effect. 55% of animals entered the full remission. We think this is also an additional proof of concept of our hypothesis. And we will be very happy to additionally collaborate with the scientific community to prove our mechanism of action, and also to achieve our current goal is to do a placebo-controlled phase 2A trial. So thank you.
Thanks very much, Daniel, and also the whole team, for presenting this really innovative and novel approach. It's kind of interesting that these drugs were, have been around for a long time, but just in this particular context, they seem to be creating some real progress in the treatment of type 1. I had a question for you, and then I also want to invite others in the room to ask questions, but I guess maybe, any of you can answer this, but do you think that your therapy is targeting an endotype? So for example, the company Diamid, they created the GAD vaccine, and they demonstrated high efficacy in a predefined genetic subgroup of T1D patients in the phase 2B clinical trial. And would your team hypothesize a predefined genetic subgroup might respond most robustly? So for instance, like the Diamid subgroup was composed of the HLA DR3 DQ2 haplotype, which is really 50% of all T1D patients. So what is your thought about endotypes and whether or not this therapy might be, really, really helpful for certain endotypes?
Thank you, Monica, for an interesting question. I'll attempt to answer it. I think certainly there is much room for research in this area. And what we have found, obviously, there is clinical evidence that there is a significant dysfunction in terms of the DPP4 serum expression, which is probably caused by some genetic markers that have not been evaluated yet. And I think it's a very exciting way to look at the diagnosis of type 1D and to actually be able to diagnose the disease before it's in full onset. And certainly the therapy that we have here, the triple therapy, has been around and has been used to, double combinations to kind of target certain aspects of this disease. However, now that we see the effect of the triple combination, it will be exciting to see which markers specifically are targeted. And this is why, as Daniel said, we're really hoping to collaborate with the scientific community and get the support of the labs that are in some brilliant minds in the academia that would be able to pinpoint the exact markers and maybe see the connections that are not evident to us as of today, but maybe become evident in the nearest future that would help to prevent type 1 diabetes from, its onset, but also to help people with mature type 1 diabetes through perhaps an elevated version of the treatment that we have shown you today. I hope that answers it.
Yeah, no, it did in some ways, but yeah, it does appear that we've had several conversations with scientists really pointing to the fact that endotypes are really something to be considered. So yeah, probably something for a conversation when you engage with potential funders and other collaborators. And I wanted to open it up to the audience. I see several people from industry and as well as from academia here. So if you'd like to throw a question in the chat or ask a question yourself, please do so. You can unmute yourself.
Hey, thanks so much for a great presentation to all the team at Levicure. This seems like a very scalable potential solution to type 1. What's next for Levicure? What's your path forward? Do you have some sort of a Gantt chart for the next couple of years, next couple of steps?
Great question.
Unfortunately, I don't think I have heard the question. Would you please repeat it again? Because I think I was cut off from the sound.
Oh, yes, of course. What is next for Levicure? What do you have a Gantt chart or a path forward or next steps over the next few years?
Certainly, I think Daniel can take it away because he is leading the strategic development of the team.
So basically, our next step for sure, our main goal for is to conduct phase 2A placebo to the whole world that it works in the way we imagine.
I'm sorry, Daniel, you were a little bit garbled there. Can you repeat your answer?
So do you hear me well now?
Yes.
So our main goal now, which we have, is to finance and conduct a phase 2A trial, placebo-controlled. So this is our main goal, and for sure our roadmap is currently that we're publishing the data which we have obtained with the Professor Levit inside his clinics, the NOG data, the mechanism of action, and going to the regulators to get additional approval for the trial and get the finance either through the VC or through funding or through the grants. So this is currently our status, and our main goal is for sure to conduct the phase 2A trial.
Well, that seems very good, like a really a great next step. We'll look forward for the paper to come out, and keep an eye on your team as you progress. I think it's a very interesting first set of data, and hopefully those who are listening today can reach out directly to you if they have further questions. So thank you again to Levicure, Daniel, Lucy, and Dr. Levit, and we really wish you the best as you continue to develop this therapy.
Thank you so much, Monica, for having us and for your support. And thank you for everyone who found the time to attend. It's been a privilege to present to you today. And I think we'd all like to thank you again and wish you a wonderful day going ahead. We'll be looking forward to hearing from you. Thank you.
Yes, we'll definitely be in touch. Thanks again, and have a great rest of your evening in Tel Aviv. Bye-bye.